In this session, we are going to study all about pollen grains. So, pollen grains are male gametophytes that are produced by microsporogenesis, which we studied in detail in the previous class. So, PMC divides by meiosis to produce four haploid microspores, which are also called pollen grains. The pollen grain which is produced is spherical in shape and about 25 to 30 micrometer in size. Each microspore or pollen grain contains an nucleus and a vacuole. So, in this microspore or pollen grain, it is covered by an intine and externally covered by an exine. So, where there is no exine, that space is called germ pore. Exine, outermost layer exine, innermost layer intine and vacuole. So, many number of vacuoles will be present and an haploid nucleus. So, this is a structure of a typical pollen grain at its formation, when it is at its formation. So, now we will have to study at the time of release how their structure will be there. So, in the very next, let us see. So, this pollen grain undergoes a symmetrical division where the spindle fibers are asymmetrical spindle fibers. Okay, it undergoes mitotic division to form an asymmetrical spindle which divides the pollen grain into two cells. One is spindle shaped generative cell and the other one is vegetative cell. This vegetative cell is large, secretes or produces intine and forms pollen tube and nourishes the generative cell. So, this vegetative cell, what it is doing? It is large in size compared to generative cell, produces intine, forms pollen tube and also nourishes the generative cell. So, coming with the generative cell, it is small, floats in the cytoplasm of vegetative cell, forms male gamete. So, that is the important. So, as I told, the vegetative cell secretes the intine. This is the innermost layer of pollen grain, that is intine. And the outer one is formed by tapetal cells. the outermost. In previous session we studied about the exine and intine. So just what is exine? Exine is an outermost and secreted by the tapetal cells. Where exine is absent, that region is called germ pore. It is through this germ pore the water, nutrients and all enter and helps the embryo or it is at this germ pore the moisture, nutrients enter into the pollen grain and forms the pollen tube. So here how many cells we are finding? 2 cells. One is vegetative cell and the another one is generative cell, and this stage is called 2-celled stage. 2-celled stage. So majority of pollen grains of the flowering plants are released. About 60% of pollen grains, 60% of flowering plants release pollen grains at 2-celled stage. That is one generative cell and one vegetative cell. Further, this cell can undergo one more division, only generative cell. Okay, only generative cell undergo one more division to form an 3-celled stage. Okay, 3-celled stage. Okay, so how many cells are here present? Here 2 cells, 1 and 2. This is 3-celled stage. 3-celled stage. Our NCERT is saying 60% of the flowering plants is releasing pollen grains at 2-cell stage. Remaining. So remaining means what would be the percentage? 40%, right? Remaining percentage is 40%. So 40% of flowering plants is releasing the pollen grains at 3-celled stage. 3-celled stage. Okay, and you should also notice that whether the pollen grain is released at 2-cell stage or 3-cell stage. If I consider 2-cell stage compulsorily, it will undergo the 3-cell stage by a simple mitosis in generative cell. So only generative cell is undergoing the division. So when I speak about 3-cell stage, here also there is one vegetative cell and 2 male gametes So when 2-cell stage falls on the stigma, they will also undergo 3-celled stage compulsorily. It is not that only one gamete is released. They want 2 male gametes that has to be released into the embryo sac. So this is with the structure of the pollen grain, and as we see, the exine is highly sculptural, or we can say ornamental, and each species have their own type of pollen grains. For example, if we see Vinca rosea that we use in Vinca rosea, the texture of the surface of the pollen grain is smooth. Whereas if we take the hibiscus Rosa sinensis, is variably sculptured. Okay, sculptured or ornamental. Okay, so that is 2 examples which I gave for the exine. So more to study with exine and intine, just we will go with. So if we take the exine as a layer, it is discontinuous layer And where the layer is absent, okay, exine absent spot, okay, that spot is called germ pore. Germ pore. So exine is secreted by tapetal cells. Tapetal cells, and the content of this exine is sporopollenin, which is an indestructible organic matter, organic material. So it cannot be destroyed by extreme temperature or high temperature, okay, extreme temperature and strong acid or strong alkali. Nothing can break the sporopollenin. Okay, so but how can they easily form the pollen tube on the stigma? It is because of the germ pore. So germ pore allows the entry of water, vitamins and boron, okay, so which is very important for pollen tube formation. It is a micronutrient required for plants, okay, so that can enter and then only we can see the pollen tube formation. Fine. Next, coming with the intine, okay, intine is continuous formed by vegetative cell involved in formation of pollen tube, and the wall of this intine is made up of cellulose and pectin. Okay, that is with the exine and intine. Next, pollen grains, as soon as they are formed, okay, so there is a rupturing of anther walls, along the line of dehiscence. So what is dehiscence? It is the breaking of wall due to hydration. So the mature anther, okay, releases pollen grains by dehiscence. Now, in the mature anther, if we consider, there is only epidermis and endothecium. Okay, so other cells, middle layer and all other tapetal cells become dead. Okay, so that is one thing.